Hey, thanks. So it's always wonderful to come back to Berkeley. I did my PhD here, and my first theoretical neuroscience class was taken with Bruno and Fritz. So anyway, it's great to come back here, see the amazing new buildings, and all the progress Berkeley has made. So yeah, I'm going to talk about emergent elasticity in the neural code for space. So this is joint work. This is a series of papers. This is a series of papers. Great. There's a series of papers and joint work with several talented people. So Kia Hardcastle is a graduate student jointly advised between me and Lisa Giacomo. Sam Oko is a theorist in my lab, and Malcolm Campbell is an experimentalist in Lisa's lab. And all of this work is a very close collaboration with Lisa Giacomo, without whom none of this work would have been possible. We're very lucky to have her at Stanford. So what are the key questions we'd like to address? We'd like to try to understand, how do we know where we are in an environment? And what does it even mean to know or learn a map of the environment? And in particular, how can known mechanisms of local synaptic plasticity learn a map of a new environment over time through exploration? Okay. So there's an area in the brain, the medial entorhinal cortex, which has neurons that have very high-level abstract information about the environment. For example, this area has border cells that fire if and only if the mouse is at a border. This is a heat map of the firing rate of a single cell. Red means high firing, blue means low firing. And these are the positions that a mouse can explore in an open field. And so this is a border cell that fires if and only if the mouse is at a border. It has speed cells whose firing rate increases monotonically with speed. It has the famous grid cells that fire if and only if the mouse is at the vertices of a hexagonal grid. And the MEC also has head direction cells that fire if and only if the angular position of the head of the mouse is in a certain narrow range. But it's important to realize that these abstract quantities like speed, position, and head direction are not directly available at the sensory periphery of the animal. Instead, these abstract representations, internal representations of space, have to be constructed from much lower level signals coming, say, from the retina, which tells you information about the visual position of landmarks and also provides optic flow that can be used in the computation of velocity. This information also has to be integrated with low-level proprioception that tells you how your body is moving through space, and also efferent motor commands that your brain sent in the past to tell your body how to move. So information has to be fused between these low-level signals to construct these high-level abstract variables like velocity and position. Okay? So we'd like to understand how this computation arises, and we're going to focus for most of the talk on the highest level of the computation, which is how you integrate information about landmarks and past velocity, which you can then integrate to finally compute position. Right? So any spatial map-forming circuit must obey a fundamental consistency condition that relates path integration to landmark-based error correction. So imagine, for example, you're moving in an environment and you see landmark A. Okay? So that triggers a pattern of neural activity, pattern A. And then you walk from landmark A to landmark B. So then path integration of velocity evolves this neural activity pattern to a new pattern B prime. Now let's say that instead you just directly saw landmark B, and that elicits a neural activity pattern B. Self-consistency means that the activity pattern B must equal the activity pattern B prime obtained from first seeing landmark A and then traveling to landmark B. So the question is, again, how can known mechanisms of neural dynamics and synaptic plasticity solve this self-consistency condition in that's required of any spatial map-forming circuit? Okay? So here's the outline of the talk. We'll first show that landmark correction is indeed something that occurs. We'll provide evidence that the stability of grid cells does arise due to continuous correction by encounters with landmarks. Okay? Then we'll develop an analytically solvable attractor model augmented with plastic synaptic inputs from landmark cells that can learn a consistent environmental map through exploration. We'll moreover analyze this model mathematically through model reduction techniques and show that this model yields a sort of emergent elasticity where landmark cell synapses behave like particles connected by springs whose rest length is equal to the physical length separating the firing fields of the landmarks. So then the system elastically relaxes to solve the self-consistency condition that I mentioned. And this model is predictive. It makes experimental predictions that are testable. It shows that grid firing fields have a subtle history dependence in their firing patterns. It explains the observation of deformed grid firing fields in irregular boxes. And it actually suggests new experiments. It suggests the potential for discovering topological defects in grid fields. And then we'll go further and look at new experiments involving virtual reality that alter the very relationship between velocity and position to make path integration and landmarks disagree. And then we probe the response of the internal representation of space in the mouse's brain when you create inconsistent conditions. And probing the mouse's internal representations in such inconsistent conditions gives us clues as to how this internal representation is constructed from low-level signals. And we'll have a model for that. So let's jump in. First, so this is an example grid cell. The basic question is, does error accumulate in this grid cell system, and if so, how? And is it corrected by landmarks? So most often you look at a grid cell just by constructing a composite histogram of where the mouse was when the cell fired, right? Or basically the occupancy condition firing rate of the cell, okay? But we could look at a finer resolution analysis where we look at the individual spikes that occur at every single position conditioned upon time since the last border touch. So here's an example where a mouse left a border and went on a trajectory, right? And these are the individual spikes that occurred at different times along this trajectory. For each spike, we compute the distance to the nearest grid firing field, okay? And we normalize that distance by the width of that firing field, okay? So this is the normalized spike distance metric. And we compute that distance as a function of time since the last border touch, okay? And what we find is that...
Is this in a dark room so that once the animal leaves the border, it has no idea where it is?
No, it's in a normal room with the usual distal cues and so on and so forth. So there could be some error correction due to distal cues, but as you'll see, there's a strong dominant effect due to landmark correction from the borders that can be detected even in the presence of any further potential correction. Okay, so here's for a single trajectory, here's the normalized spike distance as a function of time. And you can see that it increases almost by a factor of 2. So remember, 1 is about the width of 1 away from the firing field center. Within 40 seconds, right? So this tells you that potentially these grid cells rapidly decohere in the absence of landmark correction within a time scale of about 40 seconds, okay? This is the summary data for all the cells, and you see that it grows over time. So basically, the stability of this beautiful grid cell firing pattern is likely not to arise from highly accurate path integration of velocity, but likely arises from continuous correction with landmarks, okay? In this case, the boundaries for this particular environment. Okay, can we characterize the nature of this error? So there's two possibilities for the growth of this spike distance metric. One is that firing fields are just becoming more diffuse over time, right? Or two, there might be, according to attractor-based models or theoretical models of these grid cells, there might be some coherent drift in a certain direction for each of these cells. Now, these cells were individually recorded. We don't have large-scale population recordings. So a question is, using individual cell data, can we discriminate between this option or this option, right? It turns out you can if you think about it a little bit. So what you can do is these two options make very different predictions about where the spikes will occur as a function of the angle between entry and exit to the grid field. So for example, if you have an individual grid field and you just mark with a +1 if the spikes occurred outside the grid field, and with a -1 if they occurred inside the grid field, and you take the product of these things, if there's an isotropic expansion, the product of the entry and exit spiking event indicator will be flat. It won't depend on the angle of entry and exit. But if there's a coherent shift, then if the angle is small, the product will be positive. but if the angle is large, the product will be negative, okay? So we can just plot this product as a function of angle and average over cells. And interestingly, so this is the...
What are the ones and minus ones?
Yeah, so I didn't want to get too much into the details of this, but basically, the one indicates whether a spike occurred outside the center of the grid field upon either entry and exit, and a -1 indicates if no spike occurred just outside the grid field. But without going into details, this particular experimental signature indicates a positive to negative transition as a function of angle between entry to exit for the coherent drift, but a flat condition for the field expansion. And we find in the experimental data a significant slope in this thing, indicating that there's a strong contribution from coherent drift to the error, okay? So we can rule out one possible option. Another thing, also further evidence that the boundaries are correcting the error, is if you just think about it, if you hit a boundary, you receive positional information. Let's say you hit the west boundary or east boundary, you receive positional information perpendicular to the boundary, but you don't receive any positional information as to where you are parallel to the boundary. So if you resolve the error into the parallel and perpendicular components, you should see preferential correction of errors in the perpendicular direction to the last border touch, but not necessarily parallel to the last border touch. And again, without going into details, this is what the bar graph tells you, that errors are preferentially corrected perpendicular to the last border touch relative to parallel to the last border touch. Okay, so now given that there is indeed strong evidence for landmark correction in the grid cell code, let's try to model or come up with an analytically solvable attractor model for this phenomenon, okay? So the basic idea is we're going to follow past work and just assume the existence of a, let's say we're modeling grid cells in one dimension. So we're just going to assume the existence of an attractor network where you can imagine the cells are placed on an abstract ring and there's local excitation and long-range inhibition, okay? The structure of local excitation long-range inhibition yields a manifold of 1D stable activity patterns or attractor states corresponding to bumps of activity. We can summarize the state of the attractor network, the steady states of the attractor network, by the phase of the attractor network, which is by definition the position of the peak of the bump on this ring. So the entire attractor state can be parameterized by a single number, the phase, okay? Now, how do we get this system to path integrate? We're again going to follow an old idea set forth by Samsonovich and McNaughton and augmented to 2 dimensions by Burak and Feet, which is basically you have these conjunctive position and velocity cells whose efferent synapses onto the attractor network are shifted such that if a cell that is selective for eastward velocity, eastward motion of the mouse, has synapses that are shifted clockwise so that whenever the mouse is moving east, the shifted synapses will cause the bump to flow clockwise. And conversely, whenever the mouse is moving west, the west conjunctive velocity cells have synapses shifted the opposite way, so they'll cause the bump to flow counterclockwise, okay? So you can set this up in such a way that the rate of advance of the attractor phase is proportional to the velocity of the mouse. And so then what happens is, as the mouse is moving along one direction, the bump moves round and round the attractor phase. So if you're an electrophysiologist sitting outside and recording one cell, you'll see that cell's firing rate oscillate as a function of the position of the animal. So that's like a 1D grid cell. And other cells will also be similar 1D grid cells with the same period, but a shifted phase, okay? So that's like one module of a grid network in one dimension. Okay, so again, these are old ideas. What we're adding, so this is the basic idea, is the mouse moves, the attractor phase moves. Okay, now what we're adding is the influence of landmarks. We assume that a landmark cell is a sensory-driven cell, for example, a border cell, or more generally, any cell that fires if and only if the mouse is at the location of a particular landmark. Now we assume that this cell has efferent synapses onto the attractor network, such that when everything is learned, what they do is this excess excitation pins the attractor phase to the learned pinning phase of this landmark cell, where the pinning phase is by definition the location of peak synaptic input from the landmark cell to the attractor network, okay? So basically, when this landmark cell is active, the phase of the attractor network will become pinned to the pinning phase of the landmark cell. Okay? Now, of course, none of this solves the self-consistency condition I mentioned yet, because what might happen is the landmark cell likes to pin the attractor network at a certain phase. But when the mouse approaches the firing field of the landmark cell, the attractor phase may not be at the pinning phase of the landmark cell. So this is an inconsistency condition. And so what we're going to use is we're going to use Hebbian plasticity of these landmark cell synapses in such a way that one would hope that if there is this inconsistency condition, then the landmark cell synapses will learn to shift their synaptic strength to the location of the phase where the attractor network is, okay? And we can do that using Hebbian learning, okay? So I've been giving you just pictures to explain everything intuitively, but don't worry, there are equations underlying all this. But also don't worry even further, because I won't show them again, all right? Yeah.
It seems backwards. Shouldn't the sensory input have a lot more information about where the animal should be? Looks like you're adjusting the...
Yeah. So there's an adjustable parameter, which is the strength of landmark cell input. And that can be adjusted, and there's a range over which it works. What's really interesting is that there should be even higher order regulation that can adjust that parameter as the reliability of path integration and landmarks vary, right? For example, you can destroy landmarks, and path integration will work in the absence of landmarks just fine for a certain amount of time. According to our data, about 40 seconds, right? So we do have that parameter, the strength of landmark cell inputs. Okay, so this is the full, these are the full neural dynamics equations. So this is, it's kind of like a neural field model where S of U is the firing rate of a cell at position U on the ring. It's just a standard nonlinear neural network dynamics with a translation invariant connectivity that has a local excitation long-range inhibition. And then Wi of U is the synaptic strength from landmark cells to landmark cell I to an attractor cell at position U on the neural ring. And this operates on a slower timescale t, and it's basically just Hebbian associative learning with weight decay, okay? So this represents a complicated infinite dimensional dynamics of neurons and synapses, where neurons are evolving according to their own neuronal dynamics that are affected by the synaptic dynamics, and the landmark cell synapses undergo Hebbian dynamics. So we'd like to understand this complex coupled interplay between neurons and synapses, and you can do this basically by model reduction. Okay, in a situation, I neglected the landmark cell input, but there's additional landmark cell input here to the dynamics of the neurons. But in a situation where the landmark cell inputs are weak enough that they don't change the shape of the attractor bump, but just cause it to flow, you can summarize the entire neuronal dynamics again by a single number, the attractor phase of the landmark. It evolves due to path integration, but then the pinning phase of the landmark cell also plays a role, such that whenever the position of the mouse is within the firing field of the landmark cell, then, and this is the parameter, Peter, that you were alluding to, the strength of landmark cell inputs. But whenever the landmark cell is firing, then there's a pinning force that pulls the attractor phase to the pinning phase of the landmark cell. And again, because the bump shape isn't changing, Hebbian plasticity will learn to make the efferent synapses onto the attractor network some superposition of bump states that is itself a bump state. And you can summarize the synaptic strength of an entire landmark cell's synapses using a single number, the pinning phase. And so basic model reduction of these equations yield a set of finite dimensional equations that we can intuitively understand. So here's a situation where the landmark phase is evolving due to path integration and being pinned by the pinning phases of the landmark fields, but the pinning phases of the landmark fields themselves are evolving to learn the average attractor phase that occurs whenever the landmark cell is firing, okay?
So, Surya, are there multiple grid scales here?
You can add multiple grid scales.
You don't really need grid cells. You can have place cells, you can have pretty much anything, right?
Yeah.
It's easier with grid cells, but...
It's easier with grid cells. You don't necessarily need grid cells to do this. That's absolutely true. You do need some kind of, in this framework at least, you need a one-dimensional manifold of stable activity patterns.
2D, right?
We'll generalize to 2D next, yeah. Yeah?
So what happens in this
It breaks down if the velocity changes extremely rapidly.
What does it mean extreme? Can one actually do an experiment with the running mouse or put it in virtual reality and break?
So what happens is over the range of velocities we see that this model sort of works. But what happens is in most experiments where the mouse does a rapid movement, it's going from one end of the border to another, one border to another, so then the border would just correct it, right? So you'd have to do sort of controlled experiments where there are zero landmarks, look at variable ranges of velocities, and see if the grid cell code decoheres more rapidly than you'd expect under faster velocities and slower velocities. And no one's done that controlled experiment, to my knowledge.
This is an obvious, in some sense, prediction of this type of model, putting the mouse in a virtual reality and letting it run rotating things quicker or slower, right?
So we'll talk about a virtual reality experiment at the end. It's not this exact same experiment, but yeah. I mean, in Ila's models, for example, she also sees that if the velocity is too fast, then you get slippage in the attractor mechanism. Right? Yeah. I'll show you. Yeah. I'll show you. They're all coupled. That's how we're going to get to an elastic system of landmarks. So to look at Ilya's question, imagine, so how would the system work just in a simple toy problem? Imagine a situation where the mouse is moving back and forth in a 1D environment with two landmarks. So a west border cell and an east border cell, say. So what does the dynamics of learning look like in the reduced model? So first of all, the mouse starts off with the west landmark, and let's say the landmark cells are pretty strong. So they pin the attractor phase to the pinning phase of the west border cell, right? Then path integration advances the the attractor phase, okay, until it gets to the east border cell. Now this is before learning, so there's nothing guaranteeing that the final attractor phase after path integration equals the pinning phase of the east border cell, okay? So then what will happen is the east border cell will pin the attractor phase to the pinning phase of the east border cell, but also the east border cell synapses will be plastic. So the pinning phase will move slightly closer to the phase of the attractor network, okay? Then again, path integration will retard the phase back to something else here, but again, before learning, there's no guarantee that the pinning phase equals the attractor phase once the mouse gets here. But pinning will move the attractor closer to the pinning phase, and plasticity will move the pinning phase closer to the attractor phase. Okay, and after several cycles of exploration, you'll be at this happy equilibrium where the phase advance due to path integration equals exactly the phase difference between the pinning phase of the west border and the pinning phase of the east border, okay? And vice versa, okay? And in this happy situation, all of the phases of the attractor network assigned to different positions on the track become independent of which border cell you came from, okay? So for example, in the middle, you get the same phase regardless of whether you're coming from the left or the right, okay? So that's kind of what the learning process looks like. Can we say something more general and rigorous about it? We can. So one way to think about it is to take the attractor phase, which is fundamentally a circular variable, and unroll it. Think about unrolled phase to make it a linear variable. If you do that, then you can think of the landmark cell synapses, or more precisely their pinning phases, as points sitting in unrolled phase. And you can equate unrolled phase with physical space, right? Again, modulo the grid spacing, okay? So here are two landmark cell synapses. They're like particles sitting in unrolled phase. And before learning, you start at the location, the attractor phase starts at the location of one of the landmark cell particles, and it advances, but it overshoots the other landmark cell particle. And so then there's a force that pushes this landmark cell particle out, and vice versa. If you start here and go to here, you know, according to this picture, you overshot. So the attractor phase overshoots, so there's a force pushing this landmark cell out, okay? Until you'll reach an equilibrium where, again, the distance between the landmark cell synapses in unrolled phase equals the phase advance due to path integration. Or equivalently, if you think about, equate unrolled phase with physical position, the landmark cell particles have a distance equal to the physical distance between the landmark cell firing fields outside in the world. Okay? So you can think of this as two landmark cell particles connected by a damped spring, right? And if the two landmark cell particles are closer to each other than the actual physical distance between their firing fields, the spring is under compression, and they'll expand and move apart. On the other hand, if they're further apart, the spring is under tension, and they'll come closer together. Okay? So that's an intuitive characterization of what was going on in this learning dynamics. But we have a mathematical proof that this is true for arbitrary environments in d dimensions for arbitrary numbers of landmarks. We'll restrict ourselves to d equals 1 or 2. I was a string theorist at Berkeley, so I'm willing to go up to 11, but recently I haven't been doing that. Yeah.
Noise in your path integration, or
Yeah, you can add noise, and that it kind of averages out over the noise.
you want to, right?
Huh?
You want to, otherwise, to be consistent with experiments, you need noise, right?
Yeah. So you can add noise to it, and it slows down the learning a little bit, but it's robust to that. Because you're always doing this. It's like adding noise to a spring particle system, right? There's this relaxational dynamics that gets you to this equilibrium state. And if you add noise, you just fluctuate about the equilibrium a little bit. Okay.
So you've got a way of taking two ground truth references and essentially self-generating a linear interpolation between them.
Yeah.
Right? And it sounds like you're reusing that same interpolator over here and over here, so you don't have to have a separate one, or do you? Does this interpolator only work between these two grid cells? Or
I'm not sure I fully understand your question. We're basically, what we're doing is we're setting up an attractor network with external inputs from landmark cells, such that the synapses of the external inputs from landmark cells learn to pin the attractor network at exactly the phase the attractor network would arrive at if path integration took it there. That's all we're doing. Okay? We can generalize everything to 2 dimensions. So now the neural ring becomes a neural sheet or a torus. Again, this idea has been used in previous work on grid cells, where basically you have local excitation and long-range inhibition. And in this situation, Turing pattern formation takes over, and the stable states of the system are these hexagonal firing patterns on this neural sheet. Okay? So the set of all possible stable states are parameterized, for example, by this unit cell, which is a twisted torus, and the set of stable points are stable activity patterns are parameterized by points on the twisted torus. So then as the animal moves around, the grid on the neural ring will move around, and if you're a neurophysiologist listening to one neuron, it will give you a grid-like firing field. So again, these are the states of the 2D attractor network. As the animal moves from one end of the environment to another, the grid firing pattern translates, and so this is the set of attractor states that you see. And then if you have a border cell that's coactive with the grid network as the animal is moving from point A to point B, the border cell will connect to all the grid cells that were active during this motion, which is this pattern. And then in the twisted torus, what that does is it sets up a 2D attraction mechanism such that the attractor state, whenever that border cell fires, will be attracted down in this way, but doesn't feel a net force along the other direction. So everything generalized to 2Ds in ways that you'd sort of intuitively expect. But now you have to think about corrections in multiple dimensions. Okay? Okay, so now that we've generalized to 2D, we can make further connections to experiments. The first one is the following. It's an interesting prediction, actually. So we analyze a situation where you have a 1D environment with 2 borders, a west border and east border, and then we found sort of path independence of the internal representation of space. But imagine, for example, that you also have a south border cell. Okay? So then, according to the equivalent particle spring picture, you have the west border cell synapses, east border cell synapses, and south border cell synapses. There are 3 particles. They're connected by springs whose rest length is equal to the physical separation between each of the borders. Because there are pathways that go from the... Oh, by the way, one thing I didn't mention, what is the spring constant of the springs in this equivalent system? It's related to the frequency with which the mouse goes from point A to point B, or landmark A to landmark B. So this picture takes into account not only the geometry of space, but the statistics with which space is explored, as you'd expect, because plasticity picks up on statistics in general. Okay? So now again, when you have the south border cell, what happens is the south border cell is also connected to the east and west border cell through springs. So what happens is this extra interaction from the south border cell doesn't allow the equilibrium separation of these particles to be equal to the rest length of the spring. What happens is the equilibrium configuration actually leads to some compression in the spring, which means that the phase advance due to path integration actually overshoots the phase difference between the pinning phases of the landmarks. So now you should get some history dependence in the grid cell firing field. And if you work it all out, what it predicts is the following. If you compute grid cell firing fields but conditioned upon the last border touch, you should see that the grid cells are shifted towards the direction of the last border touch. Okay, there's a few steps of logic that lead to that prediction, but that's the prediction. And furthermore, if you condition on trajectories in which the mouse leaves the west border, it'll be the grid cell firing fields will be shifted little bit west relative to the other option of conditioning on the east border. Okay, these shifts are pretty small. According to their model, their order of magnitude 1 to 2 centimeters. So it can be a little difficult to find these small shifts, especially when grid cells are noisy. So we looked for these shifts in a very large data set collected from Lisa Giacomo's lab. Yeah?
Does the shift rely on learning, or is it?
In a fully learned environment, the shift will be present.
But I
Yeah.
thought why did the springs compress? I thought everything was consistent. That's part I didn't.
So that's true. Everything is consistent if there's only 2 border cells. But when there's a third border cell, I mean, everything's consistent 3-way, but not pairwise, right? Like, think about it this way, right? If this spring will not be able to settle into its rest length because there's extra tension contributed from these 2 springs.
Well, unless they're at the equilibrium, right? Then there's no tension.
Okay. The equilibrium length of this spring is not the same as it would be if you erased this interaction. That's all I'm saying.
Think about it.
In any case, hopefully you understand the experimental prediction, which is very simple, just a slight shift of the grid cell firing fields, even in a fully learned environment with no deformations, towards the direction of the last border touch. Okay. So again, it's a small shift. So we looked at a large data set of about 800 cells from Lisa Giacomo's lab. And the way you can detect such a shift is to cross-correlate, say, the west-conditioned border touches, the firing fields conditioned on west border touches, against the firing fields conditioned on east border touches up to some spatial lag, and compute how much you have to shift the west-conditioned firing fields, say, east to match the east-conditioned firing fields. And what we find, so this is the cross-correlation between west and east-conditioned firing fields, according to the spatial lag with which you shift the west-conditioned firing fields as a function of space. And you find that you have to shift them east to achieve maximal correlation and not really north or south. Okay? Similarly, we can ask how much do we have to shift the south-conditioned firing fields to make them maximally match the north-conditioned firing fields. And we find that we have to shift the south-conditioned firing fields north in order to get a maximal match. And the shift is about 2 centimeters as predicted. Okay? There's some related work by Vijay Balasubramaniam and colleagues who also modeled attractor networks and found a similar shift in deformed environments. Okay. So there's another famous result that these grid cells deform if you put them into an irregular container, right? And there's actually, believe it or not, phenomenological models where these individual grid cell firing fields are like particles with interatomic potentials with repulsion and attraction. And it turns out they predict the data, but there's no mechanistic basis for thinking about individual grid cell firing particles as atoms that repulse and attract each other. But it turns out in this alternate emergent elasticity framework, where the landmark cell synapses are pulling and tugging on the grid cell attractor phase, you actually get this, in this mechanistic framework, you actually get these bends. So this is experimental data, and this is simulated data from our model. And it's very easy to understand intuitively why these bends are coming. A border cell that fires along this border will tend to pin the attractor phase in the two-dimensional twisted torus along a certain manifold of phases, right? And then the position at which the mouse's grid cell next fires depends on moving a certain distance away from the border. And that distance will evolve depending on where you are on the border. So basically you get these grid cell firing, these bending patterns here. You can also visualize the bending by looking at the phase assigned by the attractor network as a function of physical position. Okay? All right. So here's part of theoretical neuroscience is not only to explain old experiments, but maybe to suggest new experiments, right? So here's an interesting experiment. It turns out that both of these grid cell patterns are stable, okay? This firing pattern has 3 grid cell firing fields on top and 3 on the bottom, but this only has 2 on the top and 4 on the bottom, right? So there's a topological defect here. Why is it stable? So it boils down again to winding number. Remember, the consistency condition is, let's say that you have border cells here and here. The phase advance due to path integration better equal the phase difference between the pinning phases, right? So you could achieve this consistency condition with a winding number of 1 here, or a winding number of 2 here, right? Both of them are equally consistent, and it turns out they're both stable under the equations. But how do you create the topological defect? Okay? Well, you can create it through a sequence of deformations, and I'll go through this quickly. Basically, you start off with an environment where there is no topological defect. You kill off a chunk, and there are these rich landmarks that continuously pin the attractor. So you chop off one corner, and then you stretch it out, right? The attractor phase will be dragged with the pinning phases of the landmarks, and then you close it back so this border cell has a chance to also exert a pinning phase here. And it turns out this will be perfectly stable, right? So this is an experimental protocol for introducing topological defects in grid cells, which is a completely new class of experiments that I've never seen before. And I hope somebody will do it. Lisa and I have other things we're working on, so we're not going to do it, actually. So that I hope that entices other people to do it. Okay, so now let's go further and actually try to probe how these internal representations are constructed even further. And a great way to do that is to just alter the laws of physics, right? We'd like to alter the laws of physics and then see how the neural representations respond to these altered laws of physics. And the way we're going to do that, the way this was done in Lisa's lab, is through these virtual reality experiments where you alter the very relation between distance moved as measured by velocity and the degree of advance of the world as measured by vision, okay? And we can model this, and we find that our model yields a dynamical phase transition in which grid cells can break free of landmarks and remap, okay? So again, we're probing this final correction stage between how landmarks and velocity signals are fused to construct an internal neural representation of position, okay? So here's here's the basic idea. Here's the mouse. It's going to be walking on a ball. This is an experimental setup developed in David Tank's lab. And as it walks along the ball, the world will move forward according to how fast the mouse is moving on the ball. So hopefully this works. Okay, there you go. So the mouse is moving fast. The world is moving fast. The mouse stops. The world stops moving, and so on. Okay? So this is a baseline condition where the mouse gets experience with this virtual reality environment. Okay? And so what Malcolm in Lisa's lab did was he measured grid cells and border cells both in a 2D environment to identify that they really were bona fide grid cells and border cells and speed cells. And then he looked at those same exact cells in the virtual reality environment in the same recording session. And he found that grid cells in the real world behave like grid cells in virtual reality. Border cells in the real world behave like border cells in the VR in the sense that their firing rate was tied to landmarks. And speed cells also behave like speed cells, so that's great. The mouse kind of thinks the virtual world is a real world, all right? Or at least the mouse's medial entorhinal cortex thinks that. I don't know what the mouse is thinking. I wish we knew. That would solve a lot of problems in neuroscience. Okay, now we can control the system. So we can change the rate of advance of the visual scene in response to how much the mouse moves. So for example, so this is like a baseline condition where the mouse is moving at some velocity, and this is the rate of advance of the world, okay? So we could consider a gain increase where for the same number of steps of the mouse, the world moves much faster, or we could, so this is, gain decrease, the world moves slower. So you can see for the same velocity of the mouse, the world is moving slower. Or we could do a gain increase where for the same velocity of the mouse, the world moves faster, okay? So now the question is, how does medial entorhinal cortex respond to these changes? Now, whenever we do a gain increase or a gain decrease, we do this in blocks of just 5 trials, right? Interspersed with long periods of the baseline condition. So we do not allow the neural representation to learn. We probe the response of the neural representation in response to a sudden perturbation and then take it away, okay? So as usual with all of these things, the data came first and the model came, and the data didn't make sense. So we had to come up with a model to understand the data. The model made predictions and so on, which helped us to analyze the data. But I'm a theorist, so I'm going to present the model first. So how would we model this thing? Again, we're going to look at the same attractor network whose phase advances due to path integration, presumably driven by both optic flow signals and by locomotion of the legs of the mouse, okay? But at the same time, it's a Q-rich environment, so there's a continuous sequence of landmark cells that are active in sequence. So initially, when the mouse approaches one landmark, that will have one pinning phase. And then as the mouse moves forward, it'll approach another landmark, it'll have another pinning phase, and so on. So there's a sequence of pinning phases marching forward in time as the mouse moves. And also the attractor phase is going forward in time, partially driven by path integration of velocity, but also driven by the pinning phases. So you can model this coupled dynamics between path integration and landmarks literally as a system of coupled oscillators, okay? So basically, the phase of the of the attractor network, again, advances due to path integration. The landmarks advance, again, due to the velocity of the mouse or internal estimate of the velocity of the mouse at some rate, okay? But that's just the advance of landmarks, of visual landmarks. And then the landmark pinning will drag the attractor phase. So this is literally a Kuramoto oscillator model, okay? And we know a lot about coupled oscillators. So when the whole system is settled and learned, what will happen is in the baseline condition, the phase advance of the path integrator, the red arrows, will always equal the pinning phase of the attractor network, and everything will be fine. Sensory cues and velocity cues are all in register, and everything's fine. We model the gain increase or gain decrease by a change in one of these constants, in Ka, for example. What that does is it changes the frequency of one of the oscillators. And we know a lot about coupled oscillators with different frequencies, right? If the frequency difference isn't too large, then what will happen is you'll be in a subcritical situation where, let's say so this is baseline where landmarks and path integration agree. In a subcritical gain change or a small gain change, what will happen is there'll be a slight mismatch in frequencies, so the attractor will, say, lag the landmarks, but they'll move in register with each other. There'll be a constant phase lag. What that leads to is if you plot grid cells as a function of position on the visual track, there'll be a phase lag between the gain change condition and the baseline condition. Okay, and that can be detected by a shift in the cross correlation between firing patterns before and after the gain change. However, if the frequency difference is too large, the coupled oscillators cannot entrain each other, and essentially what happens is the attractor phase can break free of the landmark pinning phases and go in a complicated dynamics. So essentially when visual cues and proprioceptive cues mismatch, when they mismatch to a great extent, the path integrator just breaks free, and then what will happen is the grid cells will remap, okay? And so there'll be a drop in the cross correlation between baseline and gain change, okay? And so just to show this, so this is a situation where everything matches. This is a situation where, say, for example, in a gain increase, this is a weak mismatch and you get a phase shift in the attractor network. This is a strong mismatch and you get a decoupling between the attractor and the landmarks. And so you get remapping in the grid cell network. Okay, and we did Lisa and Malcolm did a weak gain increase computation. I'm kind of running out of time, so I'll make a long story short. Under a weak gain increase, we did indeed see a phase difference between the baseline and gain change condition. And this is the cross correlation. There's a peak at a certain phase lag. And we can make all of this quantitative. In a strong gain decrease condition, we saw destruction in the cross correlation. And we saw remapping when you saw a strong gain change. Intriguingly, border cells were unaffected, indicating that border cells are primarily driven by visual inputs and they ignore proprioception. And there's some interesting effects of landmark friction that we can talk about. And so here's the basic idea. There are these two regimes predicted by theory. Under a weak gain change, you should get a phase shift in the neural representation. Under a strong gain change, you should get a remapping, and we see both of them. And now it can end. So again, this is a sequence of work, some published, some under review. And again, I'd love to thank extremely talented collaborators, Kia Hardcastle, Sam Oko, Malcolm. And again, this is a very close collaboration between my lab and Lisa's lab. And as I mentioned, none of this work would have been possible without Lisa. And we're super happy to have her at Stanford. All right, thanks.
In the wild, walls are not as common, so I would imagine that the landmarks are going to be things like trees or rocks.
Yeah, exactly.
And how do you see that playing out in terms of, like, somehow the system has to kind of tag something like a landmark, right, attach a cell to it. It has to happen fairly dynamically and quickly.
Yeah, so that comes through the visual system or any other system that can detect fixed objects, right? So, entorhinal cortex gets strong inputs from all sensory areas. So if there's any stable sensory pattern that can be elicited by a sensory system, and that stable neural activity pattern has connections onto the attractor system, and there's Hebbian learning, then the whole story goes through, right? Like any reliable relationship between attractor phase and the sensory cell will be reinforced in the synapses from the sensory cell to the attractor network. So then when that sensory cell is next fired, it'll pin it back to the phase that was there when it first encountered the landmark. Yeah.
Do you need the entorhinal cortex to navigate around the world? That was only
There's interesting experiments on that. Lesions of this area can affect learning in environment, but sometimes they don't affect the ability to navigate a well-learned environment. And
if you throw them in a non-well-learned environment, it's not like it's hard to say, right? They're not going to bump into walls.
So not bumping into walls is a low bar for navigation. Yeah, it's an interesting question. Like, no one has done the killer experiment to show that these brain areas are obligatorily related to navigation in a well-learned environment. That's difficult to show because there could be multiple brain regions involved.
HM could wander around the world pretty well, right?
HM could wander around the world. I don't know if he could learn his way around a new environment he'd never seen before. I mean, he couldn't remember a conversation 5 minutes later, right? So it'd be unlikely that he could navigate or learn to navigate a new environment. He could navigate his childhood home, like childhood areas, like really well, which indicates that, as we know, there's transfer of information from hippocampal areas to cortical areas. So which brain regions contribute to navigation is a very complicated story. Yeah.
Are there experiments where the visual input is removed in the animal?
Yeah, so people have done lots of experiments completely in the dark, and place cells and grid cells persist for some amount of time. People quibble about those experiments because there could be olfactory cues and other things that are not destroyed, and so on and so forth. But the consensus in the field is that you don't necessarily need sensory inputs for the stability of these place cells and grid cells for short periods of time.
You wouldn't affect the landmark input?
Well, presumably, if the landmarks are driven by visual inputs and you get rid of visual inputs, you no longer have active landmark cells.
So would you see that in the recording?
Oh, one thing I didn't tell you is that in this work, in the virtual reality work, we also looked at how is velocity constructed, right? Because in this virtual reality environment, you have two sources of information about velocity. One is optic flow, like just forget landmarks, how fast or how large is the optic flow? And two, how fast are your feet moving? And it turns out we can dissect the differential contribution of these two signals to the speed tuning, both at a behavioral level and a physiological level. And we found that as the mouse is moving fast, it pays more attention to optic flow, both in physiology and behavior. And when it's moving slower, it pays more attention to proprioception, both at the level of physiology and behavior. So the brain, I think, uses every signal available to it to construct an internal representation. And when the reliability of different signals vary, it ignores the unreliable ones and pays more attention to the reliable ones. Yeah, Eric?
Forgive me if I missed this, but do you see a role for plasticity in the velocity integrating synapses as well as the landmark hitting synapses?
Yeah, definitely. So there are several effects that might be explained through changes in the gain of the velocity cells, right? So yeah, that's a possibility. We didn't need it to explain the effects that we looked at, but of course it's a possibility in more general scenarios. Maybe Wolfgang? Yeah.
I guess if one takes a closer look at the neurophysiology of adrenal cortex, one sees all kinds of local dynamic difficulties, like neurons having different firing rates. You have shorter plasticity of synapses and probably some history dependence. So can we understand that a beautiful theory can somehow work for such a crappy implementation of a system?
I'm not sure what the question is.
So you have a beautiful theory like theory of physics, right?
Yeah.
Maps apparently to reality because the physical laws are of a similar kind of clean nature.
Yeah.
If we look at neurophysiology,
Yeah.
probably see lots of details. Every neuron has a different baseline firing rate, right?
Yeah.
Plasticity, you have different inhibitors.
Yeah.
Which don't fit to this theory, right?
Yeah.
But apparently there are some ways how this network organization principles can override this local kind of diversity.
Yeah. So we kind of modeled an idealized system and that idealized model was enough to make experimentally testable predictions which were then verifiable. We're of course not addressing issues about the robustness of this model to various perturbations. So there must be some mechanisms for ensuring this robustness in the face of intrinsic variability in the level of synapses and intrinsic excitability of cells and so on and so forth. Presumably, I mean, there's many cells that are involved, right? So actually, one thing we did look at was pairwise correlations between grid cells and border cells, and they were weak, which suggests that a grid cell is presumably integrating from many speed cells, which means if there's fluctuations in the strength of synapses from speed cells to grid cells, those fluctuations must be being averaged out, right? So you could have a lot of heterogeneity at a single cell level, but if the system is fundamentally operating in a distributed way, that's an easy mechanism for attaining robustness. Whether that is the mechanism or not, I don't know, but we didn't need to model any of that to get interesting predictions.
We're going to move on just because we're
Okay, sorry.
we can save the rest for the break.